I think the future is quite bright for gene therapies for neuromuscular disorders. We have approvals for spinal muscular atrophy, and we have a potential approval for Duchenne muscular dystrophy, as well as other gene therapies in development for Duchenne muscular dystrophy. I think it's plausible that there will be multiple gene therapies approved for Duchenne muscular dystrophy in the future. And there are also gene therapies under development for limb-girdle muscular dystrophy. It's important to have complex care facilities administer these gene therapies, and newborn screening in particular will play an important role in identifying patients who are eligible for these therapies and routing them towards facilities where this type of intervention is possible. I think it's exciting and important that different gene therapy options are being developed for specific diseases such as Duchenne muscular dystrophy. There may be certain patients who respond better to one gene therapy approach versus another, and there are likely to be both advantages and disadvantages to a particular type of gene therapy. This problem is especially thorny for Duchenne muscular dystrophy because the gene is so large. There's just no way that the entire gene will fit within an AAV vector, and thus the the approach that many investigators have taken is to try to figure out what are the most important pieces of the dystrophin gene that we need to insert in this AAV vector. And so the different approaches that are being developed for gene therapy for Duchenne muscular dystrophy have a lot of similarities. There are a lot of similarities between the genetic material that is inserted in these AAV vectors, but there are also differences because there are certain regions that there's a consensus, yes, these regions are really critical, we can't leave these out. But then there are other regions where it's not really as clear how important this one versus that one is. And so there are some subtle differences between the various gene therapy approaches for Duchenne muscular dystrophy. And there are also thoughts about developing ways to deliver the entire dystrophin gene. If that is feasible, then that holds a lot of promise. But but with every approach, there will be pros and cons. So I think that it will be important, ideally, for there to be multiple approaches available for patients, and different approaches can be tailored to a specific patient's situation.
Head-to-head comparison in this space is extraordinarily difficult. As you can imagine, it's a highly heterogeneous disease to begin with in terms of each individual's experience of the disorder. And then every trial has its own unique inclusion criteria, baseline population, way that they're assessing their patients in follow-up. Some have muscle biopsy data, others have muscle imaging data, others might assess those things, but at different times, others might not assess those things. So to try and go across programs becomes extraordinarily challenging to come up with precise, balanced, statistically informed ways of doing that. And another thing that's nice to see in the space is that there's collaborations around some of the important topics, like safety, that there's been a collaboration between all of the different sponsors sharing their information regarding adverse events and what might be leading to them. But this allows us to learn about things that are leading to improved tailoring of this approach to the population. We learn things like maybe certain genotypes are more at risk of having a reaction, and therefore we should be either giving people more immune suppression if they have certain genotypes, or they may not be able to receive the therapy safely at all, and therefore should be excluded from some approaches. The different programs have different viral vectors they use, and that may be an advantage. If somebody is seropositive to one, they may be seronegative to another viral vector, although there's a lot of cross-reactivity between these different adeno-associated virus-related vectors, and therefore positivity to one may predict positivity to another, and again lead to the need for either immune suppression or a different approach for those patients to stabilize their disease.